That diversity of sponsors reflects kind of the diversity of Mark's interests and behavior. That Mark got his undergrad degree at Cornell, and then went on to do a master's at Cambridge, and then completed his PhD at Princeton under Henry Bourne, and now is out here as a Miller Fellow. Mark particularly wanted me to point out that his office is downstairs by the foot of the T-Rex in 1120 DLSB, and I think he's feeling lonely, right? Maybe want some visitors down there to talk to him about what he's up to.
No, it's because that's a vertebrate, and this is a talk about invertebrates. So.
I was getting to it, yeah. So, you know, if you've seen the title for today's talk, you've probably figured out that there are invertebrates involved, which you may wonder, you know, perhaps.
Alrighty, so thanks for coming out today. Yeah, so this talk is going to be about the animal that you see up there. And given that most of you are vertebrate zoologists, you know, an invertebrate is an invertebrate, so you probably don't recognize what that is. But that is a hermit crab. And so this talk will explore how these little animals find food in a social context. And the things that they're finding, both food and shells, are essential resources for them. They're effectively treasure. And so the title of this talk, How to Find Treasure on Tropical Beaches, has that meaning, but it also has another meaning. And the other meaning is that this system is in itself a treasure, and I sort of found it a little bit accidentally. So I started off as a biologist studying vertebrates, and I turned to the dark side and began studying invertebrates. And so the question remains, why would one do such a thing? So why would you want to study something like a hermit crab? Well, this talk will try to demonstrate the unique and really useful features that these creatures have, but I'd also like to begin with a quote that goes back almost 4 decades now in ecology that, you know, gives hermit crabs a little bit of PR. And the quote comes from a paper by a guy named Childress, and it goes as follows. For the behavioral ecologist to combine the elements of behavioral and ecological theory into one viable framework, an organism amenable to observation and manipulation, and exhibiting a wide range of behavior patterns must be found. Hermit crabs, in general, present such a highly compact natural system. They are abundant on most beaches and mudflats, and large numbers are usually concentrated in relatively small, easily accessible areas, making observations, manipulations, and collecting extremely easy. Their small size makes them excellent animals for experimental work, both in the field and laboratory, and they exhibit a wide range of interesting behavior, from the spectacular shell fights to subtle displays of dominance. In addition, their wide range of habitats, from temperate to tropical and terrestrial to benthic, make them amenable for testing a wide range of ecological theory. So hermit crabs are pretty cool, and this guy suggests that they are ranging over a number of different ecological areas, but what is that range? Is it just one or a few hermit crab species that are spread around the world? No, it is 800 of them. And so the vast majority of these are marine. These are the ones I first started studying. And there's one in the unlikely place of the Republic of Vanuatu that is a freshwater hermit crab. And then there's these. And these are what we're going to talk about today. They're very interesting ones that live their entire life on land and often go kilometers from the ocean, and they only return there just to release their eggs. And so there's only 12 species in this family. It's called the Cenobidae, and one of them you may have heard of is called the coconut or robber crab, which weighs in at 5 kilograms as the biggest terrestrial invertebrate that's known. And you don't want to mess with a coconut crab because they're well known to, first of all, have a fetish for going into people's houses and stealing pots and pans, which they like to drag out. And when you try to fend them off with a broomstick, they're well known to be able to snap that broomstick. So, I decided not to pass out a coconut crab today, but I will pass out, for those of you who have led a very deprived life as vertebrate zoologists, pass around a hermit crab. And this hermit crab is the one that we'll talk about today. It's Cenobidae compressus. And so, you know, be careful because they do pinch. They won't, you know, break a broomstick. And have a great experience getting to hold these interesting invertebrates. Okay, so as I said, I started studying mainly marine hermit crabs, and I've been doing that for a few years. And I was in the process of, you know, finishing up my thesis, and I was pining to get back into the field, and I just wanted an excuse. And the excuse was to go and study these terrestrial guys. And so I only have had 2 field seasons with them, but I'm leaving next week for my 3rd, and I hope to have many more in the future. So this talk will talk about this kind of interesting system. And so Again, this species, Cynodita compressus, and the area where I study them is in the southwestern part of Costa Rica, right on the Osa Peninsula, where there's very few tourists. It's very isolated, and it's a very wonderful place to study these animals. All right, so here's an outline for the talk. We'll go through a little bit of natural history of these crabs, and then the bulk of the talk is going to be field experiments, which really are what hermit crabs are best for. It's combining an experimental approach inside the actual context where they operate out in nature. And so we'll look at their communication, or rather their absence of communication in these terrestrial ones. We'll look at how they forage socially and how this social foraging gives rise to a sort of accidental altruism where they help others find resources. Then we'll examine how their aggregations form and dissolve. And then finally, we'll see how new homes are incorporated into their populations. And then, time pending, we'll look at a few lab experiments that are currently in progress on these critters here in Berkeley. All right, so what are you doing if you are a terrestrial hermit crab? How do you spend your day? Well, you spend your day wandering the beach looking for two things, okay? The first is food. And if you're a terrestrial hermit crab, food constitutes almost anything. So these invertebrates have one of the most diverse diets of any crustacean, and it varies from all sorts of, animal matter, from dead shore crabs, even one another. They can be cannibalistic, to human-introduced things like mayo sandwiches, to all sorts of fruits and parts of plants. And hermit crabs are not above eating shit either. They will eat horse droppings, and if you're a curious naturalist, like David Barnes was, who studied these critters in Mozambique, is he found that they could move into a location where there were no other food resources naturally available, but except for human shit, where humans were occupying that area and just getting their business done and leaving. And so these crabs could persist off of that. And remarkably, some piles of human shit could generate up to 61 crabs in an aggregation. So that's impressive. All right, so what else are you searching for? You're looking for food. The other thing you're looking for are shells. And if you're a hermit crab, you are desperately in need of a shell. So that's the unifying feature of these animals' natural history, is they have this kind of dainty little abdomen, and then needs this external shelter, one that they don't make, one that's left over from gastropods to inhabit. And it turns out that shells are extremely limited, both in the population where I study them and in all those that have been studied around the world. And so, transects, hundreds of them in this area, have found just, a few empty shells, and most of them turn out to have a rock in them or to be broken or in some way uninhabitable. And so these are very limiting. They seem to limit the population size much more so than just food. The other thing is, where can hermit crabs get these shells? Can they go and just yank a gastropod out of them? Well, it turns out that they can't. You can do experiments where you house them with gastropods, and they're pretty ineffective at doing anything like that. Also, for these terrestrial ones, you can toss them into the water and see, hey, will you, go in there and get any marine gastropods, which are the shells that they inhabit, and they'll quickly, just like Darwin's iguanas, when he would throw those in, they'll swim back and come to the shore. They don't want to be in the water. So the best spot for getting shells is pretty much a conspecific speck. And because of that, you can find hermit crabs in iffy situations, like they'll inhabit palm nut shells instead of a gastropod shell. But it gets worse. Sometimes they'll be found like this. Not sure if that's Coca-Cola or Pepsi. Hermit crab wasn't advertising the brand. But then eventually they get so desperate that it's really just, it's a style, and it's beautiful. All right, so that's some natural history, but going down there was, not just to gather natural history, but try to see something about what these crabs are doing in terms of their communication. That was the original goal. And so for several years before that, I had studied these marine hermit crabs, which perform these very prominent displays where they'll throw out their chelipeds and hold them in a direct display. And so you'll see a video of that. This crab here will be approached by another crab, and it'll perform a display. So here we go. Howdy. And then that crab will get out of there. So, what does that thing mean? What's that all about? Well, we can figure that out by consulting Mr. T from A-Team. And Mr. T had a great quote, and the quote was, "I pity the fool." Okay? And the subtlety here, though, is that Mr. T pitied the fool because Mr. T was a very strong man, and he would pity anyone that would mess with him. The difference among these hermit crabs, and kind of the interesting form of communication that's going on is they are signaling their intentions. So it doesn't matter how big a crab is. Any crab can attack, and these crabs that perform these threats seem to reliably follow through with some sort of attack after that, regardless of their size. And so that was a major focus of my thesis. So I was interested in how information in these conflict scenarios could be transmitted in situations where there was some difference in the genetic interests of these two parties. And so I was sitting around and doing writing on these things, including on those insignificant creatures that are invertebrates, or that are vertebrates, I'm sorry, like monkeys and birds. And I wanted to get to the field again. And so what I did is I said, all right, I'm going to go with a powerful theoretical framework in mind and go and ask what terrestrial hermit crabs are doing. And so with that framework, I went and I sat on the beach, and for many instances, I waited till crabs naturally cross paths. And so when marine crabs do this, they frequently threaten. They're defending their space. And so I watched several hundred circumstances like that, and I saw very few displays. And I did a number of experiments to see if they would display in controlled circumstances, and they didn't display. And they didn't display over food or anything like that. And so I was there in Costa Rica for a while, and I decided I needed to do something rather than just sit on the beach and watch these crabs not display. And so what we'll talk about are some of the field experiments that I did, in particular, what you see in that picture. And so what we'll do is take a little kind of tangent from that picture, because we'll abandon out-of-shape Americans and their metal-detecting machinery, and we'll look at a natural circumstance where organisms need to find some valuable resource, and how do they do that? All right, so if you look at hermit crabs, these terrestrial ones on the beach, you find that they're fairly spread out when they're foraging. So the distance is usually about half a meter or more, and they're wandering around looking for these food resources and for shells. And so there's an interesting way, though, that they can, without just discovering it themselves, locate these resources. And so how do they do that? Well, when you're walking down the beach, occasionally you will come across this. Okay, this is a video of these crabs in a very big aggregation, piling atop one another and tumbling around. And what you can do is you can go there and you can take each one of those crabs off and count it. And you count all the animals in these aggregations. And what you find is beneath them, usually fairly well buried, because the intertidal is an area where their food resources and the shells are constantly getting, sloughed around and covered, is there's something good. There's either an empty shell that they're going for or some type of food resource. And so when you sample those aggregations, a number of natural ones, defining that as 3 or more crabs in a very close area, all in tactile contact, you find that there's usually about 10 crabs, and it can range up to 70. And most of the time that's under some sort of food resource, though the rest of the time it's either an empty shell, or the prospect is that these crabs are just about to pull someone out of their shell. And so the interesting thing about these aggregations, if you watch them in nature, is you find that crabs will sometimes neglect totally unoccupied food that's within a meter or so of these clusters and just join the cluster, okay? And if you put out food just naturally and you wait, it takes a long time for crabs to actually be able to find that, whereas they seem to orient towards these aggregations. So that's puzzling. I mean, there are examples in the invertebrate world, actually the best examples are in the invertebrate world, of animals attracting one another to resources. These examples are the waggle dance and honeybees, or ants dragging their abdomen and laying a trail pheromone that helps others in their colony get back. But the puzzle here for the crabs is why would you do that when, in fact, you don't have nearly the same social structure as these ants or these bees? And so there's a big divide in the invertebrate world between what can be called socialism, which is exhibited in these species that have a haplodiploid system. That means they're all very closely related and they're all effectively a single group helping one another, and individualists like the hermit crabs. And so, I mean, classic work from Bert Hall-Doppler and E.O. Wilson and their student Tom Seeley has shown how these are effectively integrated units. But hermit crabs are not. So hermit crabs will eat quite literally ants and bees for breakfast. They also roam very widely. They do not seem to belong to a central colony. They don't return to the same spot. And they don't seem to have any type of kinship ties. So, oh, did someone just get bit? I'm sorry. You should have signed a release form. But their dispersal is based on being released planktonically and just floating around the ocean and ending up where they do. And so they do not seem to have the same social structure. And indeed, despite even trying, so Tom Seeley sent me 500 bee tags, which are very convenient to mark hermit crabs in the lab, but it doesn't change their behavior whatsoever. They're still very antagonistic to one another. So the question is, why would individuals apparently attract others to these sites of discovery when there's apparently no natural history that would indicate they should? So what we're going to look at in this talk on the experiments that follow is, first of all, how, at approximate level, do these crabs join together and form these aggregations? What are the cues or signals that lead them to form these? And then second of all, at an ultimate level, what exactly are the evolutionary consequences of doing this? Okay, so here's the setup, okay? What we're going to do is we're going to go out and do something very difficult. We're going to put in some little imprints on the beach to make a quadrat with some dowels. And we're going to put out 4 of those, and 2 of them are going to be controls. There's one that has nothing, and there's another that just has the materials which we will use to tether a bunch of crabs. So tethering a bunch of crabs just means tying around a little fishing line around their leg and effectively simulating an aggregation, which is something that's not very easily done with a vertebrate, but is very easily done with a hermit crab. It doesn't hurt it, and you can release it right after. Okay, so here's the setup. I would sit 5 meters away so the crabs couldn't see my movements, right at the entrance to the forest, and I would set up these quadrats. They were randomized order, and they were conducted in the morning and the evening, which is when the crabs are very active, when they can wander the beach in relative shade. Okay. And this is, again, a very isolated beach, so at most I'd see about half a person a day. Sometimes, though, I would see a boat go by, and they would be very confused because I would have my binoculars to watch these crabs, and they thought I was spying on them, and they would wave to me in the distance, but I couldn't explain that I was watching hermit crabs. All right, so the timeline was I would go out and set these quadrats, and then I would give that 10 minutes of just waiting so the crabs could return to their basic activity, and then I would start baseline recording. So from -10 to 0, I would record at every minute how many crabs were in those quadrats by going across them. Then at time 0, I'd run out there and put out these materials, okay, and see what happened, and continue recording in each minute interval how many crabs were in there. All right, so what happened? We're going to go through a set of experiments. So here's the first one, okay? 2 of them were controls. And what you see there are the number of crabs on the y-axis, and across this period from -10 to +10 minutes. And so the controls effectively had no change, okay? They remained pretty constant, and there were no aggregations that were formed within them. It was, at most, fewer than 2 crabs a plot. So what happens when you just tether a few crabs and leave them there? Effectively simulating an aggregation where there's no actual resource there. Well, you find that within just 10 minutes you can get upwards of 10 to 15 crabs in those plots. So they are lured in very quickly. Do we need to do a statistic to test for this? I love this quote. This is advice from Lord Rutherford. And Lord Rutherford said, if your experiment needs statistics, you ought to have done a better experiment. Problematically, Lord Rutherford was a physicist, and as a field biologist, we all know that things are messy, and so we need some statistics here. And the ones that we'll use are pretty simple. We'll look at a t-test that compares this interval when I would run out and put everything into the quadrats, going over each one, when I could survey directly, not just with binoculars, and then the last time period at +10 minutes, where I'd go out and hand count all the crabs. Okay, so there is a significant effect here in terms of crabs being attracted. But a hermit crab is really 2 things. A hermit crab is a crab, and it's a shell. And so what exactly, and there's one right now wandering on the table. So the question is, what exactly were these individuals attracted to? And so we can test that effectively by tethering some crabs, live crabs, or tethering some shells. Again, control plots, no difference. It doesn't matter when nothing's in there or when the materials that we use to tether these crabs are in there. But it turns out that when you put in 3 naked crabs, 4 naked crabs, that is extremely attractive. In fact, it's significantly more attractive than putting in 3 empty shells. Now, it seems actually the effect here from these empty shells being put in was only generated as follows. So crabs would come along and accidentally encounter these non-moving shells. They'd find a shell that was better than the one they currently had, and they would enter that. They would then be stuck because that shell was tethered there. And so they would start to make the movements of a simulated aggregation, which would then lure in more crabs. So it seems, therefore, that even in the absence of any resource, any food resource, or any empty shell, just having a naked crab and having them move in a spot is very attractive. Crabs seem to be lured into that site. Okay, but how? Is that chemical? Is that visual? Is it something else? What exactly is it? So here's the profoundly complex setup we need to do to test this. So what you do is you take a dowel and you stick that in a plastic cup. It's opaque so crabs can't see it, and you poke 30 holes in it, okay? What we can then do is set that up, and first of all, test the control plots. No difference, same story, okay? Doesn't matter if you have a little white cup there with a dowel in it or nothing in that plot. That doesn't attract crabs. So does it attract crabs to have crabs in that cup, or to have their actual main food resource, which is one of their most frequent in the population I study, which is beach almond? It does not. So having 3 crabs in a white cup, or having beach almond, which is freshly cut up in that cup, does not generate any aggregation. There's no difference. So how can we test another possible mechanism that may lure them to these aggregations? Something like visual cues. Well, when you're sitting around trying to figure out it might come to you in unlikely moments. And so one of the moments was a shot glass. Put some crabs in a shot glass, because in fact that was all that was available at the field station. Okay, so here's what you do. So you turn that shot glass over, and you also need to put a little plastic substrate there because crabs are going to move around and they would otherwise dig up a lot of dirt, so you can't even see what's going on inside the shot glass. Now, how do you know that no visual, no chemical cues are leaking in that case? Well, it turns out that hermit crabs are very attracted, not surprisingly, to the scent of a dead crab. The reason they're attracted to that is not just because it's a food item, but because it's an indicator that that crab was recently killed and a new shell is potentially available. And so what you can do in this circumstance is you put, just a few times, it's mean, you smash a crab and you put it under that setup. And it turns out that does not lure in any other crabs until you lift the cup, and then it does. So this effectively trapped chemical cues fairly well and only showed the actual movements of the aggregation. All right, so what happens when you do that? Well, control plots, you have your empty shot glass, all the same materials, or you have an empty plot. There's no difference, okay? But what happens when you put some crabs under a glass, 3 or 6? Well, you find there is a significant effect, but it's very, very minor, okay? Let's boost up those y-axes, and what you find is that it does actually lead crabs to come, but it doesn't start to generate these fairly large aggregations that are generated in a short time otherwise by tethering the crabs. So why is this? Well, the explanation seems to be, and I'm going to show you a piece of video that shows you an example, is crabs would come. They would be attracted from fairly far distances. They would arrive at these shot glasses, and they would circle them, trying to get into the aggregation. But after circling for long enough and not being able to get in, they would leave. There was no opportunity for a sort of positive feedback to be generated, and the crabs wouldn't even stay for very long. So it seems visual cues are sufficient, but they're not strong enough to keep crabs there. All right, so let's graph all of the conditions that had a significant effect, okay? And what do you find when you do that? Well, here's the proximate explanation. It seems that when you have an empty plot or just chemical cues alone, that's not sufficient. But when you add in visual cues that will lure crabs in, but they will only stay there if they can actually get into the big tumbling and rumbling around and have tactile contact with other crabs in that aggregation, feeling around for a resource, either a shell or food. And then finally, the single most effective condition, which is when those crabs are taken out of their shell and they're completely naked, adds to it, and it seems that maybe that's an extra potential chemical squirt of something that indicates that the crab is shellless and therefore has been recently kicked out of its shell. Okay, so those are the proximate mechanisms that crabs seem to get to these aggregations, but we still have a puzzle in mind of why, of what is going on at the ultimate level. And so, is this just a complete exploitation of cues, or is there something more subtle? Well, we can get rid of a few explanations. So first, is, and we haven't talked about this in detail, is the possibility that maybe these aggregations form and they're effectively this dilution effect, where you're less likely to get picked off by a predator in an aggregation compared to when you're alone. Well, it turns out that if you're in a shell, these crabs have essentially no risk from most predators that are on the beach. And I've seen a non-hermit crab been taken by these hawks or other birds of prey that will come down and just grab them and then just leave a nice pile of white shit after they've eaten them and fly off. And the hermit crabs are left there perfectly as they were before, whereas these other crabs, like ghost crabs, are gone. So other experiments, I'm not going to go into them, also did not support a predator-based explanation. And in fact, when you put out simulated aggregations of just these shells and you look at predator landing, you don't find any difference there. These predators don't seem to go to spots where aggregations have formed more than they would go to spots where individual crabs are. Okay, one other explanation. Let's think about maybe how could it still be mutualistic? Well, maybe it's hard to get to tear into some of these fruits or other items that the crabs eat. Well, the staple resource in their diet, which is beach almond, the crabs only consume when it's ripe, and it does not seem that there's any need to have another crab help you to tear it open. But one explanation, and this is an interesting one, is that crabs might use these big get-togethers, which are apparently around food resources most of the time, as an excuse to swap shells. And so when you're a hermit crab, again, you really need these shells to survive. Without them, you are quite literally dead meat. And what happens when one crab gets kicked out of its shell is it creates this possibility for a vacancy chain, where the next biggest crab can take over that shell, again leaving its shell, and that can cascade all the way down. So everyone, except for the one who got kicked out, could potentially benefit. Now in these experiments, they weren't carried out for very long. These were short time intervals, and I did not see any changes of shell in them. But we need to actually examine this. And so I'm going to speak about an experiment that was done last field season in 2010, which I'm not going to show you any results, but I'm going to walk you through the logic of it and also talk a little bit about the qualitative results that went with that. And so this was, again, to simply set out some quadrats and to tether 10 crabs, okay, to create a simulated aggregation of that average size that we saw in the natural history section. And over that was put very high-definition video. So I have many terabytes, actually, of data that I'm going through right now with collaborators who are capable of tracking these animals. And we've developed software which is very nice to be able to see these individual movements, which is on a great field context. It's out in nature, but it's also on a two-dimensional substrate that is very easy to track the contrast of the shells with the beach sand. Okay. So what are the things that can be gotten from this? Well, the first is, over a long-term period, what happens? Are there any shell swaps? Do crabs actually switch that? Is there a potential for benefiting? We're also, as I mentioned, and this is with Ian Cousin, who loves collective animal behavior. That is how huge swarms or aggregations of anything behave. And so with him, we've developed this software to start to track how these crabs move right around and within these aggregations. And so we can see exactly how their bias and trajectory is changed based on their distance from the aggregation and the size of the aggregation. And I'm just going to show you a quick video clip. I don't have it in the presentation because it's a very large, many gigabyte thing that I didn't cut. But this is just kind of the chaos that you see there, which can be made sense of by video tracking. And so you see one little guy who just ran in from there, and these are several tethered crabs, and these are all the other ones kind of running around and basically feeling for potentially a resource, either a shell or a food item. So we're also interested in using these experiments to think about some other questions, and one of the interesting ones is, over a long-term period, how do these aggregations build up? And so most of the data on animal aggregations have been either natural observation, so no experiments in a wild context, which is great because you have ecological validity, or in the laboratory doing very controlled experiments, but then it's a lab and it's not a natural context. And so this is kind of exciting because it's the possibility, which is what hermit crabs are good for in many ways, of combining that and doing this in the field to generate very easily aggregations based on simply tethering some animals. And so we can also address some questions. Is there an upper limit to aggregation size? Well, it turns out that a whole set of experiments were done, which take a very long time, I assure you, to tether many hundreds of crabs to one stick and leave them there. And what happens when you make such a big aggregation? Do any other crabs come to that spot? It turns out they do. There does not seem to be a limit to aggregation size in these hermit crabs, and in fact, with several hundred tethered, you will find more and more crabs coming. So within literally a quadrat that is 0.6 by 0.6, you can have many hundreds of crabs piling on top of one another, which takes a very long time to count. Okay, do these dissolve naturally? Do the crabs just go away after they don't find what they want? Well, leaving these for a long, long time, you find that no, they will stay there, and they will remain, if not build up. And so in these experiments then, after exploring that kind of length of time effect, the following was done. So I would go out to both of these quadrats, including the control, to make the same movements, and I would gather all of the crabs that had accumulated there at 1 hour into the experiment. And I would simulate a predation attack. I would take up every crab, okay? I would grab it and I would put it in a Ziploc bag really quick. That would allow me a very reliable count, and with a field assistant, we would quickly put little duct tape pieces on each crab, which is useful to see if those same crabs will return to the plot after. We would then dump out that Ziploc bag, and all this happened in just 5 minutes, and the crabs would leave. It was a very cool kind of like cellular process of just seeing them all disperse, and they would leave many meters from the site. And so what is interesting is after this, you'd find the following. You'd find that these crabs would reform an aggregation of the same or a bigger size within half of the time it took to generate. And so it seems a possible explanation is that crabs are actually attracted to a predation site. So they leave when the actual predation attack occurs, but after that, it's quite useful to come back because there could potentially be a new shell available. So the things that stimulate these aggregations seem to be unique in many ways from other animals, in that first you have a social facilitation to get to the spot and potentially swap shells, and then second of all, if there is an attack, the crabs will leave, but they will return after that very quickly, forming bigger aggregations in some cases to get at that potential new shell. All right, so I'd like to switch gears, and every kind of research program needs an outreach thing, and here's the outreach that was done here. So last field season, with many people, many dozens of people, we boiled out several thousand gastropods. Okay, we didn't care about the gastropods. We were mean to them. We cared about the hermit crabs, and we painted those with exterior house paint and individually numbered them all the way into the many of thousands. Okay, and the idea here was, I thought, wow, this is a brand new thing. It hasn't been done. This is so interesting. Let's leave these shells out here and see as the hermit crabs come in. And I'll explain what can be gotten from this experiment, but I later realized, like 2 weeks ago, when I received a request from the Journal of Archaeological Science, which I don't know anything about archaeology, to review a paper that was about basically hermit crabs coming to shell middens of prehistoric people and robbing them, and leaving their new shells and taking sometimes these very old fossil shells. So this experiment has probably been done, thousands, if not millions of times. But let me explain to you the logic here. The logic is that when you go and introduce a limiting resource into an animal population, for instance, you give birds, if you like vertebrates, strange people like vertebrates, you give birds a nest box and you say, all right, what's going to happen? Let's put out a bunch of nest boxes. What you can't know is what was their old home like. With hermit crabs, you can. So leaving all of these shells, which we did in a very small few meter by few meter quadrat, every day across a month period, we would go and exhaustively gather the shells that crabs would trade in. They would bring their old homes. They would find a new home they liked, which they had many to select from, very little social competition, and they would leave those. And so we could, in fine detail, measure various aspects of these shells. And I'm not going to present all the data. Many of it is still being analyzed, but crabs would pick homes that were twice the size of their old homes. So they wanted a more spacious home. And this is sensible in a way if you're a hermit crab, because if you're a male hermit crab, studies have shown that dominance and your access to females is determined by size. But likewise, if you're a female, it's also determined by size, your reproductive success. And the reason is that the number of eggs that you can carry around in your pleopods are determined based on the size of your shell. And so a bigger shell is going to be more optimal. The other thing that's sort of unique about introducing this limiting resource, in which has been now going on for the past year is tracking how these new homes are incorporated in the population. And again, unlike a nest box or anything else, a cavity or a cave, these homes move. And so with GPS tracking all up and down the beach, we're starting to see how these resources are getting incorporated into the crab population, which is interesting. All right, so I'd like to talk about these homes a little bit more. And home might not be the best analogy. Maybe a better analogy is a car, because these shelters, which the crabs carry around with them, and they're sort of like a Flintstones vehicle, because they're the ones generating all the movement, they last for a while, it seems. And they're traded, kind of like old used cars. But it's not clear, and it's an interesting question to ask, is how long do these shells last in the population? So with a very wonderful, smart expert on gastropods named Gary Hermit, who's at UC Davis, we've started to think about how we can get at this question. And incidentally, Hermit is a remarkable expert on shells despite being blind. And he is notorious for going into museums, and the way he recognizes shells is he kind of feels the external outside, and he feels kind of the braille pattern, and he'll go into a museum drawer and he'll just be like, you know, you have two species here. And a decade later they'll find out that, you know, they had either misclassified or they never actually did the taxonomy right, and there was really a cryptic species in there. So he's a wonderful, wonderful expert at shells. So bringing this car analogy a little bit more is we're interested in two potential ways to see how long these shells last. So with a car, of course, you have a year, a make, and a model insignia. So you can go and you find your used car in the parking lot, and you can quickly know how long it's been around. So what has been done in the prior field season, which will continue to be done, is the following. So we kind of, all that loud drilling sound, you know, stopped. But what will be done out in the field is to make a lot of loud drilling sounds, because using a portable Dremel, and without really damaging the shell, because these are very tough and hardy marine shells, is an insignia is made for each year. And so this is a very long-term study, the type of thing that, you know, it will pay off maybe like a decade from now. And you can go out and you can resample these populations and see if some of these shells with that unique insignia, which is not confounded by any sort of natural marking, are still there. But there might also be a way to get at aging shells more quickly and not waiting so long. And so one way is with cars, you have wear, right? Now some of that wear is external, but you also have an interior to a car, and that wears as well. And it turns out for hermit crabs, as a guy named Peter Abrams first figured out back in the 70s, is that for terrestrial hermit crabs, not for marine ones, there's substantial wear inside. In fact, the entire inside of the shell will also often be taken out, so that the spiral of the shell will be completely abraded, and you'll have a much bigger internal volume inside of the shell. And the shell modification, it turns out, is something that, at least it seems, crabs like to do, so it gives them extra space in the shell. And so a byproduct of that, you know, outreach program that we talked about a moment ago, where we left all those shells for the crabs, is that we can maybe start to see how much shell modification occurs after a year by going out and recollecting those same shells. And the logic would be that you can take paired shells that have the same diameter, and you can then pipette a certain amount of water in there until it reaches the brim. And you can calculate the internal volume for those shells that were put out last year and have now been in the population for a whole year, and they were quickly taken up within just a few weeks. And you can compare that difference in internal volume to brand new boiled out shells. And that gives you a potential measure of the change in internal volume over 1 year. And you can then use that to go out and take natural shells that are in the population, see how much their internal volume differs from the baseline of boiled out shells. And you can divide that figure into that and get a measure of how long those shells have been in the population. And so this does assume that there's a linear increase in internal volume, and that does need to be actually checked, but it's an interesting way to at least get a first grasp of how long these shells may last in the population, and it could be quite a while. Some gastropod experts have suggested that it may be, you know, more than a decade. All right, so I'm actually doing very well on time. so I will be able to get to these. And these are, extremely briefly, some lab experiments that are going on. And so if you went up to the 5th floor, you would find that there are many hundred terrestrial hermit crabs there. And so this space, which was the space of George Barlow originally, is actually appropriate to house hermit crabs, because I was reading his obituary recently, which was written by Steve Glickman and Roy Caldwell. And in it they quote Barlow, and he says that his earliest memory is of trying to keep hermit crabs alive in a small bowl. And so now his laboratory, which was mainly for fish, has a lot of hermit crabs in it. So in some ways, that's very appropriate. Okay, so what are some of the things that are going on in the lab? Well, one is to start to see, and I'm not an expert at all on molecular ecology, but these hermit crabs are a very interesting species to study with respect to that. And the reason is that Cynobia compressus has a distribution that extends all the way from lower California all the way down to Chile. An extremely broad distribution for an invertebrate. And so with Matt McBain, we've been just initially starting trying to examine if there's any differences in these populations in terms of their genetic structure by looking at some primers that are available for crustaceans and for hermit crabs in particular. And so we have some samples from all over the world, and hopefully we'll get more. And I didn't expect that this would go so fast, because I didn't practice this. So now with a little bit more time than would be expected, I'll end with one more slide. And this is a slide of some student projects that are going on from some undergrads who are very bright and very motivated and care about the invertebrate world, which is wonderful. And so one of these projects, which is Jennifer Bates is working on, is to see basically how crabs respond to a mirror. And it turns out there is old evidence from Brian Hazlett at the University of Michigan that hermit crabs are very responsive to mirrors. So they have very keen visual capabilities. But the experiment here is to test whether or not crabs, when you ruin or erode their shell or give them a bad shell, are more attracted to mirrors. The logic being that they're effectively trying to find another conspecific and get to that point. Another experiment that is being carried out by Marianne Davies is trying to build on this field work, which shows that hermit crabs learn the where from others. So they learn where there is a good resource, but the question is, can they learn what is a good resource? And in a kind of very simple paradigm with a tennis ball, which you can cut in half and then flip one side out, so you have one that's a nice green fuzz, and another that is kind of this rubbery tan color, putting those on opposite sides of an aquarium, and then pairing them arbitrarily with an aggregation. And the question is, after hermit crabs choose one of those sides, they show their own original preference, and then they're given an opportunity to make another choice when an aggregation is randomly paired with one of those. And the question is, are they now biased to effectively choose certain resources over others after that? So you then give them another choice after they have the aggregation test. And then finally, with marine hermit crabs at Bodega, which there are many local hermit crabs, is Alison Greger is basically exterminating some gastropods. And the logic here is that these marine hermit crabs, they do not seem to use visual cues as much as the terrestrial ones do. This is not a major feature of their environment. They're often covered in seaweed or it's very dark. But what they do use very prominently are chemical cues. And so if you smash a gastropod, this is effectively a signal of some sort that there's a new shell available. And so the logic is to see how these crabs aggregate around these resources and what exactly happens. All right, so thank you for listening, and I will thank many people, including the hermit crabs, but the person I'd like to thank the most is Roy Caldwell, who is the most gracious host and has just shared with me so many useful nuggets of natural history that it's very, very pleasing. So thank you for listening, and I'll be happy to take any questions that you might have. And the hermit crab survived everybody, so that's great.
Thanks, Jim. Questions for Mark? Shock the vertebrate world into... Yeah, Jenny.
Do you have any idea of what the immigration and emigration rates are for the hermit crab populations?
No, I do not. Vance, who was at the University of Washington, did an experiment, which is the only one that kind of attempted to document in an experimental way that shells were the limiting thing for the population. And he dumped in a remarkable number, something like 15,000 shells into a tide pool, and then he dumped in a control, which I think were like smash shells, into another one. And so it was like a sample of one, right? But he found that there were significantly more hermit crabs there a year later, but he was unable to differentiate whether that was due to their like migrating in and taking those shells, or just hanging out there more, or what. And yeah, I think you'd really have to do a very large scale, very well controlled experiment to get at that. But they're so abundant. I mean, when I was talking about the video tracking with Ian, he was like, why don't you just mark the whole population, and like then we'll have individual numbers and we can see them roaming on the beach. And I was like, Ian, there's a million. Like, how am I going to do that? So I don't know. Yeah.
I guess Damien, I guess, yeah.
So what goes on with evictions? Like, how often does it happen?
Mm-hmm.
Are there any sort of signals or things involved with that?
Yeah, signals. So the thing I didn't mention, and which could in some way have confounded that experiment that was on the quadrats initially, is that these crabs make sound, which is very interesting. And the context where they make the sound is basically like another crab is on their back, and they make this like I should have gotten some clips, but they go like it's this weird chirping thing. And so I think that I don't know if it helps get another crab off their back, but what it seems to do is it seems to have some eavesdropping function, and so it lures in other crabs to the site, and maybe they're kind of starting a conflict with the attacker who's going after their own shell. Could get that individual off their back, but it also could contribute to the buildup of aggregations. So I think there could be signals. I mean, there was also some experiments by some people in Mexico who like anesthetized hermit crabs and then like left them out there, and they could show that crabs could basically like assess weaker crabs, and even though these crabs were kind of walking around, they'd just jump on them and they would start pulling them out of their shell a lot more than other crabs. So it seems that they are assessing that, and probably the way they do that is they jump on another crab's back, and that crab kind of like tries to throw them off and makes these sounds, and if it doesn't do that as vigorously, then they'll flip it over and start reaching in there. But these things can last for so long that I don't have any data for like how successful they are, because they can, I mean, the ones that I've seen that are successful, like I just lie on the beach for like 3 hours, and it's like they're just sitting there trying to pull that crab out the whole time. So it take a long time, I think, to get those measures. But yeah, it happens.
There's another guy behind, next to Susumu. I can't see who it is. But yeah.
When you did your outreach experiments with
Right.
them, did you use one species of gastropod?
I did, yep. It was Nerita scabrocosta, and it's the most common in the hermit crab population, and prior lab-based experiments had shown that they prefer that species above all others. I think it has naturally like the biggest internal to volume weight ratio. But obviously the ones that they could turn in could be any species. It wasn't just those.
Because the sister species that's on the other side,
Mhm.
on the Caribbean side,
Yeah.
is in a greater variety of shells, and it's smaller, and then it goes into one possibility, but
Yeah.
one or two different types of shells and it gets bigger.
Okay.
So it's interesting that the ones on the Pacific side are only in that one type.
Yeah, it gets pretty big on the Pacific side, that particular shell. And I mean, I haven't identified every shell species they're in, but there's at least like 30. But I was thinking this field season, I'm just going to bring them back and just give them to Vermeer. So,
So your control with the opaque white plastic cup
Mhm.
and hole for the
Yeah.
chemical concentration,
Mhm.
I'm concerned that that's gonna even with a cup with holes,
Mhm.
is gonna significantly change both the distribution of the chemicals that
Mhm.
the chemical heat and the strength of the signal.
Mhm.
So did you try using a negative control of the dead crab in your cup and see if
No, but that is such a powerful chemical signal. Like, it's the single thing they're most strongly attracted to. It generates bigger aggregations than tethering crabs. So I would have no doubt that would lure them in. And you can even, like, I would put the dead crab after I smashed it to retain its body inside of like a Tupperware container that didn't have a lid on it, but had, it was a really high wall. And crabs would come out of the forest to this Tupperware, and so I think if anything that would have kind of constrained, chemical cues a lot more. But yeah, it certainly, I shouldn't downplay that they have no chemical orientation, because in fact in coconut crabs, which are a close relative, they've recently analyzed a lot of their ganglia structure, and it turns out that they have like an extremely enlarged olfactory bulb compared to most other invertebrates. And I do think they orient to some foods, very prominent ones, based on chemical sense. So I shouldn't say that they don't ever do that. Yeah.
Jim, did you question?
I did, yeah. It seemed like in your description of the behavior of the crabs that they're spending all their time either looking for shells or looking for food. But what's the role of reproduction? Are they not looking for mates, and how do they?
Right.
Where do they mate?
So mating is a risky endeavor when you're a hermit crab, because it means you have to come a fair amount out of your shell. And so it turns out that most of the time when they do it, it's more of a private affair. They don't want to do it on the beach. And I have never seen hermit crab sex despite watching for a long time. And so like there's not all that many descriptions of it, but they do come all the way out of their shell. And so I think a lot of the time, and these guys are not active at the hottest period of day, at midday, they'll go back under these big coconut piles. My guess is that's probably a better context to be doing that. Yeah.
Okay, the crabs or somebody's knocking, and so it's probably time to get wrap it up. But thanks, Mark.
Sure. Oh, thanks for coming. Oh, really? Yeah, I would love to. So I was neglecting the 10 or 12 you guys. So I know around. Were those I would take them, yeah. And I guess that, so I now actually have identified all the species that are there. The marine or industrial? Marine ones, the only way is literally to make a break in the shell and tickle their abdomen. The terrestrial ones, you can breathe on them and they'll come clawing out, and then you can get them. But the marine ones are, yeah, they're tough.
Yeah. I was going to say, nice job. I'll let you answer questions.
Oh, thanks for, yeah.
Yeah, hi Mark. My name is Yu.
Okay.
Very good talk. Yeah, PhD here with Robert Dudley and Dave Wake.
Oh, cool. I wanted to talk to him for a while. I haven't.
Yeah.
He does like flight and stuff.
Yeah. So my thesis is on. Is not recording. Yes, my thesis is on flight mechanics and evolution of wings and stick insects.
Stick insects. I
And
didn't
also
know they had wings. That's cool.
Yes,
Yeah.
most of them have wings.
Yeah.
Even they're wingless, external wing muscles inside. And I also study some tadpoles feeding on the surface. They also aggregate, and they.
You know what is Ian? You have talked to him, because I know. All right, he told me he was doing something with North Carolina tadpoles, and that the system was really cool because he could have a certain number of them would start swimming in circles, and they would create this vortex. Yeah.
Oh,
I don't know all the
really?
details, but he was starting to think about that in a collective behavior sense, and why
Maybe I should talk to
why they do it. Yeah.
So
yeah.
my tadpoles, they only feed on the surface of the water.
These are local guys.
Yes, they're
Yeah.
from Asia.
Oh, okay.
And
Right.
they have an oral disc, which is dorsally open, so they have unique morphology.
Yeah.
And the shape of their oral disc varies from species to species because they have different flow habitat.
Yeah.
Some are in still ponds, some are faster creeks.
Mm-hmm.
They do aggregate, and some species fasten themselves in the meniscus at the side of the creek.
Okay.
And in particularly one species, they school together in hundreds on the surface of the water.
Yeah.
And form a band, continue from where the upstream waterfalls
Yeah.
to kind of where to the downstream, but it's a big pond.
Yeah.
And that band turned out to be where the surface of vortex is. So that's a vortex created by the waterfall and bring the bottom accumulations out. So that generates not only buoyancy, also carries all the food.
That's cool.
So I've done TIV for all by using these paper pieces, and I've also tracked the tadpoles. Not the track, but video film the tadpoles.
Okay.
And
are you able to track individuals?
I haven't started tracking, but my plan is because I need to figure out the local flow field for each tadpole. And particularly for the one I mentioned, they form a band.
Yeah.
And when I saw that, I think I need to talk to you about it. Because I'll find another time to show you the videos.
Yeah, I'd love
and
to see it.
And I saw on American Scientist, the Sigma side magazine, someone was tracking the bird aggregation.
That might have been Ian.
The bird cloud.
Yeah.
Probably it's the same.
Was it starlings or starlings? No.
Probably.
Yeah, but I would totally recommend talking to him, and the reason is, like, it seems like there's overlap, and you guys could totally do something together. Because I know I think they were trying to do something where, I don't know, they put something in the water that allowed them to like better see the strength of action or how particles were moving around. But I don't know if that project ever got off the ground because they're having some difficulty like
recognizing the visions.
Yeah, basically.
Yeah.
I mean, this is a lot easier.
Really? But they have overlapping as well.
Yeah, they overlap as well, but I mean, when you generate these like, this is like 10 tethered crabs, and like most of the ones that overlap are the guys who are stuck there. And these guys that are coming in, like they'll be zipping all around, and you can track them relatively easily.
Have you so you're running the primary analysis. Do you have any results? I would say there are kind of a big set of parameters.
Yeah, I mean, we've just gotten to the point of being able to like track the movements and to have a single glob here, which is the individuals that are stuck and tethered, and to have the little ones that move. But I mean, frankly, there are so many like different relevant variables about how big that aggregation is at the moment, and about how far these animals are here. That there's like so many things we could milk from it that we need to like basically be careful to not try to do everything at once, because it's too many parameters. I think there are cool things we can get about. So another experiment I didn't mention was to have 4 quadrats. 2 of them were controls, and one of them was 3 big crabs, which were over a certain size, versus 3 small crabs. And the logic was to see what's called public information. If big crabs in a spot means there's bigger food and bigger shells for a shell cascade, and small crabs doesn't. And what we're analyzing there is something simpler about staying time, and when they get in the quadrat, how long do they actually remain there.
So they do sense things clinically.
Yeah, I think so.
So
yeah.
Oh, but they're mostly under the wing of efficiency of their aggregation.
Are the tadpoles, do they do much visual stuff? Or?
They do, yes. Because on the surface, it's very dangerous.
Yeah.
It's in the day. I will show something when I come to you. But
I would love to
the girl who asked you about the control experiment, have you talked to her?
I haven't. Who is that?
She, her name is Lindsay, and she's
Lindsay, I know her. Lindsay
Lab.
Lindsay Waldrop. Right. Yeah.
Talked to her?
I have talked to her. I didn't recognize her.
But last Friday, she just gave a talk on the bottom.
Yeah, I was on the East Coast, and I emailed her, and I was like, I was going to talk with her after to just hear what she did. But I saw her glass models of the olfactory sensing devices, which is really cool. Yeah. And I think she was saying too that she's starting some behavioral experiments with an undergrad, and she was like asking if I had any thoughts on that. But yeah, I would like to talk to her more. I'd like to see her tadpole video too.
Yeah, I will. So you're.
I forgot your name. I'm sorry.
Okay,
great.
I'll email you first.
Cool.
And you said you'll be leaving in 3 weeks.
I leave in 1 week.
Oh, you
I
leave
next Wednesday. Yeah,
so
but I'll be back. I will be back in a month. I'm only gone 1 month, and then I'm here the whole time. So
I'll email you first. Hopefully find a time before you leave.
Okay, cool. And I would also email Ian because I think he might be helpful, at least in like giving you ideas or stuff of things you could look at. Let's see. All right.
That's cool.
Cool. I guess we just leave that thing on. I'm not sure.
recording.